Okay, let's dig into this. Are you feeling maybe a bit overwhelmed by all the information swirling around about dementia?
It's a lot, definitely.
Yeah, and you want to get to the heart of the crucial breakthroughs, but, without needing a science degree.
Right.
Making sense of it all.
That's exactly what we try to do here on the deep dive. We take that complex research, sift through it, and pull out the vital stuff just for you.
And it's moving so fast, this field. Uh, Honestly, keeping up feels like a full-time job sometimes. So these focus dives are really helpful.
Totally agree. And today we're really focusing deep on blood-based biomarkers for dementia, especially Alzheimer's disease.
Hmm.
We're looking at how they're fundamentally shifting, well, everything really. Diagnosis, maybe even treatment down the line.
And our main source here is a really fantastic lecture by Professor Charlotte Teunissen. It was the Donald L. Price Memorial Lecture.
Right. And it feels important, doesn't it, to just take a moment to think about Don Price.
Oh, absolutely. His legacy in Alzheimer's research, neurodegeneration, it's immense, a true pioneer. Yeah. And not just his own discoveries, but his mentorship, too. He really shaped so many leaders in the field today.
That's right. Professor Teunissen giving this lecture, it sort of honors that whole lineage of research,
Yeah.
making the progress we're talking about feel even more significant.
Definitely. I mean, you look at his career, head of neuropathology at Hopkins, president of both the American Association of Neuropathologists and the Society for Neuroscience,
Huge role.
and over 500 papers. Plus, all the awards, Potamkin, MetLife,
Mm.
just a giant. His work really laid the groundwork for what we're discussing now.
Which brings us to Professor Teunissen's specific focus, right?
Hmm.
The blood biomarker.
Exactly. That's her main thing, developing these fluid biomarkers, mostly from blood, to get a much better handle on neurological diseases.
Better diagnosis, figuring out prognosis, tracking treatments, the whole package.
And she's not just theorizing, she runs the Amsterdam dementia cohort. That's, over 10,000 paired CSF and plasma samples?
Incredible resource.
Yeah,
Plus, she co-leads International Biomarker Network, so she's right in the thick of it.
she really is. Her work covers everything from finding potential markers using omics methods,
Right, like looking at thousands of proteins at once to see what's changing.
all the way through to making reliable tests and checking them in big clinical studies. It's about getting science to patients.
Exactly. Translating discovery into real-world benefit.
So our mission for this deep dive is pretty clear then. Understand the progress in Alzheimer's blood tests, how they're actually getting into clinics, and also explore how they might help predict who treatments will work for and if they are working.
The big hope being a faster, much less burdensome diagnostic journey for people.
Okay, let's get into it. Why are these biomarkers so important right now? What's the urgency?
Well, the urgency comes from just how tricky dementia is to manage. I mean, first off, you need early and accurate diagnosis. That's crucial.
Right, for the person, for their family, and for getting the right people into clinical trials, too.
Precisely. But it's more than just a yes or no for Alzheimer's. You also need to understand how the disease might progress for that individual. That's prognosis.
Okay, so prognostic biomarkers. What else?
Then you need predictive biomarkers, who's likely to respond well to a specific treatment. That's key for personalized medicine.
It makes sense.
And finally, when someone starts a treatment, you need monitoring biomarkers to see, is it actually doing what we hope it's doing?
So it's like a whole toolkit we need, not just one magic bullet.
Exactly. A suite of tools for different questions.
And Professor Teunissen dropped some big news early on, didn't she? Plasma biomarkers for diagnosing Alzheimer's. She said they're ready for clinical implementation now.
That's a huge statement, a really profound shift, actually.
Why so profound?
Well, think about it. Historically, diagnosis often meant invasive spinal taps for CSF, or relying heavily on cognitive tests and clinical judgment, which can take time and aren't always definitive early on.
Right.
The idea of a relatively simple blood test giving you core diagnostic information, that's genuinely revolutionary for the field.
And she singled out one specific biomarker, p-tau217, called it the absolute winner. For someone like me, what's the deal with p-tau217? Why is it so good?
What's really compelling about p-tau217 is how specifically it seems to reflect one of the main problems in Alzheimer's, the abnormal tau protein that forms tangles inside brain cells.
Okay, the tau tangles.
Right. And it seems very specific to Alzheimer's changes. Plus, it starts changing really early in the disease process, even before major symptoms sometimes.
And it's not the only one, though. She mentioned others, too.
Oh, absolutely. p-tau217 might be leading the pack right now, but there are other promising plasma markers. Some reflect the other key Alzheimer's feature, the amyloid beta plaques, and others are more general markers of neurodegeneration, brain cell damage.
So having a panel looking at a few of these together could give a clearer picture.
That's the idea, a more comprehensive diagnostic signature.
It's just wild how fast this has happened. She said the first papers were only like 5 years ago.
Yeah, roughly 5 years from first major publications to talking about clinical implementation.
That's light speed in medical research terms.
It really is astonishing progress.
So, okay, they're ready. But what does that actually mean? How do you get a biomarker like this from the lab into, like, your local doctor's office?
That's the crucial next step, implementation. And it's exactly what Professor Teunissen's group is studying right now in the Netherlands.
How are they studying it?
They're running real-world studies across different levels of healthcare GPs, specialized memory clinics, the big university hospitals.
Okay.
And they're looking at the impact. How does having this test available change the system? How does it affect doctors' decisions? And importantly, what's the patient experience? Does it lead to quicker answers?
That patient experience part feels key. Less waiting, less uncertainty.
Definitely. And they've looked specifically at introducing these tests to GPs. Interestingly, there was some initial resistance.
Resistance. Why? Seemed like a good thing.
Well, a few things came up. Some GPs worried about the practical side, adding blood draws to busy clinics.
Okay, logistics.
Yeah. And there was a clear need for really good guidance on how to interpret the results. These are new tests, after all.
Right, you get a number back. What does it mean? Especially for the patient sitting in front of you.
Exactly. But they also found things that helped adoption, like the potential for better communication with families and, of course, the main driver, getting that diagnosis faster.
So clear guidelines are essential. And what about the blood sample itself? Does it matter how it's handled?
Oh, absolutely critical. We learned this the hard way with CSF biomarkers over the years. Tiny differences in how you collect, process, or store a sample can really affect the results.
Like what kind of differences?
Things like how quickly you spin the blood down the centrifugation time or the temperature you store it at. Professor Teunissen's team and others have done tons of work on this, leading to consensus guidelines, like from the Alzheimer's Association, on exactly how to handle these samples for reliable results.
That standardization is huge if these tests are going to be used widely.
Essential for comparing results across different labs and clinics.
And she mentioned the World Health Organization getting involved too?
Yes. The WHO is working on defining what they call preferred product characteristics. Basically setting standards for what any good, widely used blood test should offer. Things like, what's its exact intended use? Who is the target population? How stable are the test chemicals? Is there good tech support and training? And crucially, is it accessible and affordable globally?
That accessibility and affordability piece feels really important. It needs to work everywhere.
Absolutely.
And who is the target population right now for these diagnostic tests? Is it everyone?
No, not widespread screening yet. As Professor Teunissen clarified, the main focus currently is on people who are already showing signs of cognitive impairment.
Okay, so people coming to the doctor with memory concerns or thinking problems.
Exactly. That aligns with the established diagnostic pathways. It's about clarifying the cause of existing symptoms.
Got it. So diagnosis is being transformed, but you mentioned these biomarkers could do more.
That's right.
Yeah.
That was the next big shift in the lecture, moving beyond just diagnosis to prediction and monitoring.
Prediction and monitoring of free med response.
Exactly. Using these fluid markers to figure out who is most likely to benefit from a specific therapy, and then tracking over time if that therapy is actually working on a biological level.
Okay, that feels like the next frontier. Personalized medicine for Alzheimer's.
It really does. The core idea is that Alzheimer's isn't biologically identical in everyone. People differ. And so their response to treatments might differ too. These biomarkers could give us a window into those individual biological differences.
So what factors did she highlight that influenced treatment response? The first one seemed pretty fundamental. The actual Alzheimer's pathology itself, amyloid and tau.
Makes sense, right? Most of the new drugs being tested directly target either amyloid or tau.
Yeah.
So logically, you first need to confirm that the person has that target pathology for the drug to even have a chance of working. That's why clinical trials are enrolling people with confirmed amyloid, for instance.
And we're seeing positive effects with those drugs now.
We are. And crucially, we're seeing the biomarkers change with treatment. Professor Teunissen gave the example of lecanemab.
One of the new amyloid drugs.
Right. And treatment with lecanemab leads to a pretty significant drop in plasma p-tau levels, and quite quickly, within about 6 months.
So the blood test shows the drug is hitting its target and reducing downstream effects like tau changes.
Precisely. It's direct biological evidence of target engagement.
And it's not just short-term effects. These markers tell us about long-term progression, too.
Seems so. She mentioned a study tracking plasma biomarkers and cognition over 15 years.
15 years.
Yeah, and they found that people whose plasma p-tau levels increased more rapidly over that time also had worse cognitive decline.
Wow, so it's not just a static snapshot. It reflects the dynamics of the disease over years.
Exactly, which is incredibly valuable.
Okay, so amyloid and tau are key. Then she talked about biological disease drivers. Sounds like going deeper.
It is. The idea here is to find markers that reflect the very earliest biological ripples of the disease, maybe even before amyloid and tau pile up significantly. And also markers that track the mechanisms actively pushing the disease forward. To do this, they use really powerful techniques like PEA proteomics.
Proteomics. Looking at proteins again.
Yeah, but on a massive scale,
Yeah.
measuring over 5,000 proteins simultaneously in the cerebrospinal fluid.
5,000. Like a molecular fingerprint of the CSF. What did they find?
Well, in an early study, they compared cognitively healthy people who did have amyloid signs in their brain versus those who didn't.
Okay, pre-symptomatic stage.
Right. And even then, with no symptoms, there were already differences in lots of proteins involved in things like immune function, how proteins are processed, fat metabolism.
So the biology is already changing under the surface.
Exactly. And they've managed to distill this down. They developed a panel of just 12 key CSF proteins reflecting these mechanisms.
A 12-marker panel. And what can that panel do?
Remarkably, it showed it could predict which individuals were more likely to progress later to mild cognitive impairment or full dementia.
Whoa. So predicting future decline based on these underlying drivers.
Potentially, yes. Identifying higher risk individuals much earlier.
Okay, this is getting really interesting. Then she introduced another layer. Molecular Alzheimer's disease subtypes. What's that about?
This is super important. It's the growing realization that Alzheimer's probably isn't just one single biological entity.
Meaning?
When they look closely at the patterns of thousands of proteins in the CSF of different people with Alzheimer's, they see distinct clusters or subtypes. Professor Teunissen's group identified 5.
5 different biological flavors of Alzheimer's based on proteins.
Kind of, yeah. Each seems to have different dominant biological mechanisms going wrong.
Can you give examples?
Sure. One subtype showed signs of what they called hyperplasticity, maybe like the brain trying too hard to adapt or repair itself. Another had strong signs of innate immune system activation. And interestingly, that immune subtype had the highest tau levels, the fastest brain shrinkage, and generally the worst prognosis.
Wow, so the immune system going haywire is linked to faster progression in that group.
It seems so. Other subtypes pointed towards problems with RNA regulation, issues with the choroid plexus, the part that makes CSF, or a leaky blood-brain barrier.
This feels huge for treatment, doesn't it?
Absolutely huge.
Because maybe an anti-inflammatory drug would work best in that immune subtype, but not so well in the others.
That's exactly the hypothesis Professor Teunissen raised. Or maybe even the current amyloid drugs work better or worse depending on the subtype. We need to investigate this.
Tailoring treatment to the specific biological subtype, that's really personalized.
But where the field needs to go.
Okay. One last factor she mentioned, co-pathologies. Other brain problems happening alongside Alzheimer's.
Right. Very common, actually. You often see things like Lewy body pathology or TDP-43 protein clumps or vascular damage in the brains of people diagnosed with Alzheimer's.
And these mess things up further.
They can definitely influence how fast the disease progresses and potentially how well treatments work.
Let's take Lewy body disease first. We have a CSF test for that already, the seed aggregation assay.
We do, the alpha-synuclein seed assay, or SAA. It's pretty good at telling you if Lewy body pathology is likely present.
Okay.
And having that alongside Alzheimer's pathology seems to be linked to faster cognitive decline. But as Professor Teunissen noted, the SAA is basically yes or no. It's binary.
Doesn't give you a level or track changes easily.
Exactly. Not ideal for monitoring treatment response, for example.
Yeah.
So they looked for something more quantitative.
Using proteomics again?
You guessed it.
Yeah.
Back to the CSF proteomics, they compared people with Lewy body dementia to others and found a protein that stood out. BDC.
DDC. Dopamine decarboxylase. Isn't that involved in making dopamine?
Precisely. And dopamine system dysfunction is a core feature of Lewy body diseases. It fits perfectly.
Okay, so they found DDC. Could they measure it reliably?
Yes, they developed sensitive assays for CSF DDC, and validation studies confirmed it. DDC levels were higher in people with Lewy body dementia compared to controls in people with Alzheimer's, even in autopsy-confirmed cases.
That's strong. And they tested this idea in a clinical trial.
They did. They looked at data from a trial for a drug targeting a pathway called PDE9 in Lewy body dementia.
Okay.
The hypothesis was, maybe the drug works better in people who have biological proof of Lewy body disease using markers like DDC.
That did it.
It looked promising. Those with the biological signature, including elevated DDC, showed better drug effects changes in brain signaling and even a trend towards better cognitive scores.
So using the biomarker helped identify potential responders. That's cool. What about DDC in blood, though? Easier than CSF?
Yeah, that's the logical question. Unfortunately, CSF DDC doesn't seem to translate well into a diagnostic plasma marker for Lewy body disease.
Ah, okay. Shame.
But interestingly, plasma DDC levels do seem to track with a dose of levodopa medication in people with Parkinson's disease.
So maybe not diagnostic for DLB, but potentially useful for monitoring treatment in Parkinson's.
Potentially, yes. A different application.
But the search for a blood test for Lewy body disease didn't stop there.
Not at all.
Yeah.
They did another big plasma proteomics study specifically looking for a Lewy body signature.
And found something.
They did. They identified several proteins like endoglin, ITG, iScan that looked promising. And some of these also helped distinguish Parkinson's from controls in another data set.
Okay.
They even developed a 7-biomarker panel from plasma that showed pretty good accuracy in telling Lewy body dementia apart from controls in Alzheimer's.
A 7-marker blood test panel for DLB. That would be a significant step forward.
Very encouraging, yes.
Yeah.
Still needs more validation, but definitely promising.
Okay, what about the other co-pathology mentioned, TDP-43?
This was really new research from a group. They found a novel marker in CSF. It's elevated in patients with a type of frontotemporal dementia, FTD, that's caused by TDP-43 pathology, compared to FTD caused by tau pathology.
Interesting. Does it show up in Alzheimer's too?
Yes, that's the key thing. It's also elevated in the CSF of some people with Alzheimer's disease.
And what does that mean if it's elevated in someone with AD?
Early data suggests that if an Alzheimer's patient tests positive for this TDP-43 marker in their CSF, they tend to have a faster rate of cognitive decline.
So it might predict faster progression in Alzheimer's.
Exactly. Another potential prognostic marker related to co-pathology.
This is all incredibly complex, but also really hopeful. Okay, let's circle back finally to monitoring treatment response.
Right. We saw lecanemab changing p-tau. Can other biomarkers track other drug effects?
Yeah. The hypothesis, she said, is that markers of core pathology, amyloid, tau, and those disease driver markers should be useful for this.
Makes sense. And she gave a first proof of concept from a trial.
The PEPIMAB trial, targeting neuroinflammation.
That's the one. It was a phase 1b/2 trial in moderate Alzheimer's, testing PEPIMAB against placebo for about a year.
And they looked at biomarkers.
Yes, in the CSF. They found that after a year, the group getting PEPIMAB had significant reductions in several relevant biomarkers compared to placebo.
Which ones?
Particularly markers related to synapses, the connections between brain cells. Things like GP43 and SNF25.
So the drug seemed to be having a positive biological effect on those crucial connections.
Exactly. It suggested the drug was engaging its target and influencing downstream biology in the intended direction. A really important proof of principle for using biomarkers to monitor treatment effects beyond just amyloid and tau.
Okay, so pulling this all together from our deep dive, it really feels like we're in a new era.
I think that's fair to say.
These plasma biomarkers for diagnosis, they're not science fiction anymore. They're here, or very nearly here, offering a way to diagnose Alzheimer's that's less invasive, potentially faster.
A huge relief for many patients and families, potentially.
Yeah. But it's not just diagnosis, is it? The really exciting future is using these fluid markers, blood, CSF, to predict who will respond to treatments.
Getting the right drug to the right patient.
And then monitoring closely to see if it's actually working biologically.
Absolutely. But it also means embracing the complexity.
Right. What Professor Teunissen really emphasized.
Yeah. It's not just amyloid and tau. We have to think about those biological subtypes, the co-pathologies. Fluid biomarkers are going to be essential tools for navigating all of that.
And none of this happens in a vacuum. It takes huge teams, international collaboration.
Definitely. Researchers like Professor Teunissen and countless others working together. It's a massive effort.
So looking forward, these biomarkers are clearly central. Which leads me to a final thought for you. our listener.
Hmm.
This field is moving incredibly fast. As we learn more and more about the biological details of dementia using these markers, we get closer to truly personalized treatments.
Tailoring therapy based on biology, not just symptoms.
Exactly. So, what might that really mean for individuals, for families, if we could choose therapies based on someone's unique molecular profile? Think about the possibilities, but maybe also the challenges as this next chapter unfolds.